From the deserts of Saudi Arabia in September 2012, an intriguing case of a viral infection was reported. It involved a man who was admitted to a hospital of Jeddah in June of the same year with pneumonia and an acute kidney injury. Only a few days later, a separate report appeared of an almost identical virus detected in a second patient with acute respiratory syndrome and acute kidney injury. The second patient initially developed symptoms in Qatar but had traveled to Saudi Arabia before he became ill and then sought care in the United Kingdom. At first, these cases were supposedly diagnosed as of severe acute respiratory syndrome, SARS, because they displayed high resemblance, but later this virus was named as novel coronavirus because the patients were much more burdened by the pre-existing health problems and were much more likely to die of their illness than SARS patients. This novel coronavirus was initially termed as human coronavirus Erasmus Medical Center 2012, but later renamed as Middle East Respiratory Syndrome Coronavirus MERS CoV. This previously unknown coronavirus was first isolated and identified by Egyptian virologist Dr. Ali Muhammad Zaki from the man's lungs. Dr. Zaki then published his findings on 24 September 2012. The isolated cells showed cytopathic effects CPE in the form of rounding and syncytia formation. In an interview on Asking, Dr. Ali Muhammad Zaki said, "I could consistently reproduce the pan-coronavirus positive result, but it was not SARS. This was the first evidence that I could be dealing with a novel human coronavirus that had not been described before." It was initially called human coronavirus EMC in a paper by its discoverer, the Egyptian microbiologist Ali Muhammad Zaki and Ron Fouchier of Erasmus MC in the Netherlands, enlisted by Zaki to help characterize the virus. Since then, a plethora of other names has been used. The paper's authors write, "After careful consideration and broad consultation, the CSG has decided to call the new coronavirus the Middle East Respiratory Syndrome Coronavirus, MERS CoV. This name is endorsed by the discoverers of the virus and other researchers that pioneered MERS CoV studies by the World Health Organization and by Saudi Ministry of Health. We strongly recommend the use of this name in scientific and other communications." A second case was found in September 2012. A 49-year-old male living in Qatar presented with similar flu symptoms, and a sequence of the virus was nearly identical to that of the first case. In November 2012, similar cases appeared in Qatar and Saudi Arabia. Additional cases were noted with deaths associated, and rapid research and monitoring of this novel coronavirus began. It spread to 26 countries. As of July 2015, a total of 1,401 patients were infected with this virus, of which 543 died. Even though the transmission rate is comparatively slow when compared to SARS-CoV, the MERS CoV infection continues to grow. Coronaviruses, members of the Coronaviridae family and the Coronavirinae subfamily, are a diverse group of large enveloped positive stranded RNA viruses that cause respiratory and enteric diseases in humans and other animals. These coronaviruses are divided into 4 genera: alpha, beta, gamma, and delta, and are classified into distinct species by host range, antigenic relationships, and genomic organization. Coronaviruses typically have narrow host ranges and are fastidious in cell culture. MERS CoV belong to the genus Beta and are genetically subgrouped into lineages C. These viruses cause a range of diseases in humans and animals, ranging from the common cold to diseases like SARS and MERS, which cause serious illness. The exact source and mode of transmission of MERS CoV to human beings is unknown. Initial investigation suggested that MERS CoV originated in bats, as the sequences related to MERS CoV have been found in several bat species. Other studies supporting a bat source showed that a bat coronavirus, HKU4, and MERS CoV can each use both human and bat dipeptidyl peptidase 4 as host cell receptors. However, MERS CoV has never been isolated from bats, so whether direct or indirect transmission from bats to people is important remains unknown. Several other animal species in Arabian Peninsula have been assessed for serological evidence of MERS CoV infection. Serological assays have improved and sensitivity and specificity have increased. In an early study, 100% of dromedary camels in Oman and 14% in the Canary Islands of Spain were positive for anti-MERS CoV antibodies. Subsequent studies confirmed a high rate of seropositivity in camels in the Arabian Peninsula and showed no evidence of infection of cows, goats, or sheep. One key question is whether MERS CoV newly emerged into camel or human populations, or whether the infection had been present for many years but was not detected. A retrospective analysis of stored human samples obtained in 2012 from blood donors and abattoir workers in Saudi Arabia showed no evidence of MERS CoV seroreactivity. By contrast, anti-MERS CoV antibodies were detected in archived serum samples from dromedary camels obtained in Saudi Arabia in 1993 and the United Arab Emirates in 2003. Furthermore, many camels in Saudi Arabia are imported from East Africa. Additional studies showed that serum samples from camels in East, West, and North Africa were positive for MERS CoV as early as 1992, indicating widespread circulation of MERS CoV in camel populations for many years. Transmission from camels has been linked to human MERS CoV infection. Although few patients have a history of camel exposure and transmission is inefficient, however, interpretation of this lack of an obvious exposure might be confounded by less direct exposure. The transmission is primarily person to person or from another intermediate host, and that some camel infections result from person to camel transmission. Additional intermediate hosts might be involved. MERS CoV particles as seen by negative stain electron microscopy. These virions are made up of a nucleocapsid surrounded by an envelope which has characteristic little club-like projections or protein spikes emanating from the viral membrane called peplomers. Virions are spherical to pleomorphic and the surface projections are evenly dispersed and spaced widely apart, covering the surface, forming a prominent fringe responsible for attachment to cells, hemagglutination, and membrane fusion. The core of the nucleocapsid in MERS consists of a 27 to 33 kilobases long chain of linear, single-stranded positive-sense RNA nucleotides. Coronaviruses have the largest RNA genome. Based on the assembly, the genome size of MERS was estimated to be non-segmented 30,108 nucleotides. An overview of the entry and replication process of MERS CoV in the infected host cell is shown here. The S-glycoprotein located on the surface of the MERS CoV virion interacts with functional receptor DPP4 to facilitate viral entry into the host. The S protein consists of a globular S1 domain at the N-terminal region, an S2 domain with two heptad repeats, HR1 and HR2, and a transmembrane domain. The S1 domain determines cell tropism and receptor interaction, whereas membrane-fusing mediators have been identified within the S2 domain. MERS CoV binds to DPP4 through a receptor binding domain (RBD) located in the S1 subunit. Subsequently, protease cleavage of the S protein leads to virus cell fusion and the release of viral genomic RNA into the host cytoplasm. The initial translation begins in open reading frame 1A and continues in open reading frame 1B after a frame shift, thereby producing the polyproteins 1A and 1AB respectively. Open reading frame 1AB, which comprises 2/3 of the genome, is responsible for encoding known structural proteins, whereas the remaining 1/3 of the genome encodes structural proteins E, N, S, and M, and 5 accessory proteins. The virus-encoded proteases PL-Pro and 3CL-Pro cleave the PP1A and PP1AB proteins at 3 and 11 different sites respectively, resulting in 16 mature non-structural proteins. The proteins involved in replication and transcription, like RNA-dependent RNA polymerase and helicase encoded by open reading frame 1AB, form replication transcription complexes. These complexes assemble at the perinuclear regions and associate with double membrane vesicles derived from the endoplasmic reticulum. It has been confirmed that double membrane vesicles are not separate vesicles, but are instead part of a reticulovesicular system of altered ER membranes. The genomic RNA contains adenylate uridylate-rich sequences called transcription regulation sequences that are about 10 nucleotides long. These sequences divide the genomic RNA into different body elements of various lengths. These transcription regulation sequences are either recognized by the replication transcription complexes to generate discontinued, short, negative-strand RNA of subgenomic length for the transcription of accessory and structural proteins, or continuous, full-length, minor-strand template RNA of genomic length for replication. The newly synthesized genomic RNAs are encapsulated in the N proteins in the cytoplasm and then transported to the ER-Golgi intermediate compartment for further assembly. The proteins S, M, and E are inserted into the membrane of the rough ER and are subsequently transported to the ER-Golgi intermediate compartment, where they interact with the N proteins and assemble into particles. The budded vesicles are then transported to the cell surface for release after maturation in Golgi bodies. In the life cycle of RNA viruses, interferons or IFNs have a crucial role in the antiviral defense and are activated by the double-stranded RNA generated during viral replication. Pattern recognition receptors, including toll-like receptors or TLRs, and retinoic acid-inducible gene I or RIGI-like receptors, and melanoma differentiation associated protein 5 (MDA5) have an essential role in innate immunity. Among TLRs, TLR3 specifically recognizes the double-stranded RNA through its ectodomain, whereas RIGI and MDA5 also recognize double-stranded RNA through their helicase domain. After sensing double-stranded RNA, the activated immune response induces IFNs and cytokines to block viral replication. To await this cellular immune response, MERS CoV4A protein binds to the double-stranded RNA and blocks the induction of type 1 IFN. In its most severe form, MERS causes an acute, highly lethal pneumonia. Renal dysfunction or failure is common in patients and could be a consequence of either hypoxic damage or direct infection of the kidney. The latter is a possibility because DPP4 is expressed at high levels in the kidney. Analysis of patient tissue samples is needed for a greater understanding of sites of infection. Unfortunately, no tissue samples are available, largely for cultural and religious reasons, so that much of our understanding of pathogenesis comes from studies of experimentally infected animals. Several animals can be experimentally infected with MERS CoV, including macaques, marmosets, and camels. Mice transgenic for human DPP4 develop severe clinical disease after MERS CoV challenge, but these mice also develop encephalitis, compromising their utility. Little is known about what constitutes a protective immune response in patients who recover. MERS CoV elicits attenuated innate immune responses with delayed pro-inflammatory cytokine induction in cell culture and in vivo, which could contribute to a dysregulated immune response. Thus, vaccines that result in only antibody responses will probably be useful in the short term, but might not provide long-term protection against MERS CoV. The clinical manifestations of MERS CoV infection range from asymptomatic infection to severe pneumonia with acute respiratory distress syndrome, septic shock, and multi-organ failure resulting in death. MERS patients who died in the first wave in 2013 were older and more likely to have severe disease requiring admission to hospital than were the secondary cases. Only secondary cases had mild disease or asymptomatic infection. Based on the data related to human-to-human transmission in several clusters, the incubation period has been estimated as more than 5 days, but could be as long as 2 weeks. MERS typically begins with fever, cough, chills, sore throat, myalgia, and arthralgia, followed by dyspnea and rapid progression to pneumonia within the first week, often requiring ventilatory and other organ support. Although most patients with symptomatic disease have respiratory illness, the immunocompromised patients can have fever, chills, and diarrhea and later develop pneumonia. Risk factors for development of severe disease in addition to an immunocompromised state include obesity, diabetes, cardiac disease, and lung disease. As lower respiratory tract specimens such as bronchoalveolar lavage fluid, sputum, and tracheal aspirates contain the highest viral loads, they should be collected whenever possible. MERS can be confirmed by detection of viral nucleic acid or by serology. The presence of viral nucleic acid can be confirmed either by positive real-time reverse transcription (PCR) on at least 2 specific genomic targets or by a single positive target with sequencing of a second positive PCR product. Available real-time reverse transcription PCR tests include an assay targeting RNA upstream of the E gene or UpE, and assays targeting open reading frames 1A and 1B. The assay for the UpE target is highly sensitive and is recommended for screening. The open reading frame 1A assay is of equal sensitivity. The open reading frame 1B assay is less sensitive but is useful for confirmation. These assays have not shown cross-reactivity with other respiratory viruses including human coronaviruses. Respiratory samples should be collected at least every 2 to 4 days to confirm viral clearance after 2 consecutive negative results are obtained. For confirmation of infection by antibody detection, paired serum samples should be collected 14 to 21 days apart with first taken during the first week of illness. A positive screening assay of ELISA or immunofluorescence should be followed by a confirmatory assay. Single samples might also be valuable for identification of probable cases and should be collected at least 14 days after the onset of the symptoms. Serological results should be carefully interpreted because they might be confounded by cross-reactivity against other coronaviruses. No specific drug treatment exists for MERS, and supportive treatment is the mainstay of management. Evidence-based recommendations for treatment provide the basis of decision-making in clinical settings. MERS CoV is readily inhibited by type 1 interferons in cultured cells, and IFN alpha-2b combined with ribavirin can lessen lung injury and reduce the lung titers when administered to rhesus macaques within 8 hours of virus inoculation. This combination was tested in severely ill patients, showing an improvement in survival at 14 days, but not 28 days. Several drugs inhibit MERS CoV in cell culture, including cyclosporine and mycophenolic acid. Other compounds inhibit virus replication in vitro, although whether these drugs will be useful in patients is unknown. Steroids should be used cautiously if at all, because their use was associated with worsened outcomes in patients infected with SARS-CoV during the 2002-3 epidemic. Recommendations for prevention of MERS are available from World Health Organization, the U.S. Centers for Disease Control and Prevention, and the Saudi Ministry of Health. The main infection prevention and control measures are droplet precautions, wearing a surgical mask within 1 meter of patients, and contact precautions. Droplet precautions should be added to the standard precautions when providing care to all patients with signs of acute respiratory infection. Eye protection should be used when healthcare workers care for probable or confirmed patients. Public Health England, U.S. Centers for Disease Control and Prevention, and Saudi Ministry of Health recommendations for management of known or suspected MERS CoV infection include the use of personal protective equipment such as gowns, gloves, eye protection, and respiratory protection. Patients with MERS should be placed in negative pressure rooms or in rooms in which room exhaust is filtered through high-efficiency particulate air filters. These recommendations are evidence-based and have proven to be effective in hospitals in affected countries. The virus can also be found in raw milk from infected camels. MERS CoV is stable in camel breast milk for extended period of time. Thus, pasteurization or cooking is recommended to destroy the virus. Raw urine should not be used for medicinal purposes because signs of disease are non-specific. It is not possible to know whether an animal in a farm, market, racetrack, or slaughterhouse is excreting MERS CoV without virological testing. Camel farm workers, slaughterhouse workers, market workers, veterinarians, and those handling camels at racing facilities should practice good personal hygiene, including frequent hand washing after touching the animals, avoiding touching eyes, nose, or mouth with hands, and avoiding contact with sick animals. The Saudi government issues updated health guidelines for pilgrims. Although MERS CoV did not cause severe community-acquired pneumonia in any of the 38 hospital-admitted pilgrims investigating during the 2013 Hajj, good infectious disease surveillance and control measures are essential to prevent the major outbreak of MERS during mass gatherings. The factors governing treatment outcome are still unknown. The lack of knowledge is hindering the development of drug treatment, adjunctive treatment, specific diagnostic tests, biomarkers, and vaccines. MERS CoV is not yet fully adapted to infecting people, and person-to-person transmission is not yet sufficiently efficient for pandemic potential. Further genomic studies will provide insights into the molecular characteristics, mutation rates, and virus transmission dynamics, defining factors crucial for species specificity and virulence, and enabling the discovery of new drug targets, novel drugs, diagnostic tests, and vaccines. Post-mortem and histological studies have not been available, and even limited autopsy or surgical specimens would be useful. The availability of an animal model of severe MERS CoV infection and disease will be important for understanding pathogenesis, natural history, and immune responses, and for developing the effective treatments. These studies are urgently needed because coronaviruses have high genetic mutability, and MERS CoV continues to circulate in countries in the Arabian Peninsula. Although interhuman transmission is still inefficient, health authorities, governments, and the research community should be prepared for the emergence of a MERS CoV with increased capacity for transmission and pandemic potential. No virus is known to do good, and as it is said by Sir Peter Medawar, that a virus is a piece of bad news wrapped in protein.